Alcoholic fermentation is a type of fermentation in which carbohydrates, main glucose, are converted into ethanol and carbon dioxide molecules. It is caused by many yeasts and some mold fungi. It refers to the chemical process in which microorganisms produce alcohol through the fermentation process. Alcoholic fermentation is also known as fermentation of ethanol or ethylic fermentation. It is generally considered an anaerobic process. It is widely used in alcoholic beverages, fuels, food processing, etc. Certain bacteria, such as Z. mobilis, can also be used for ethanol fermentation. Looking interested? Come go more in depth. Alcoholic fermentation is carried out mainly by Saccharomyces cerevisiae yeast, which is the common yeast in bread or beer, which converts 90% of the sugar into equimolar amounts of alcohol and CO2. Raw materials are sucroid substances such as cane sugar, beets, and fruit juices. The main raw material is molasses. Conditions for alcoholic fermentation. Sugar concentration 10 to 18%. pH between 4 and 4.5. Microorganism Saccharomyces cerevisiae. Absence of O2 and the presence of phosphates. Fermentation temperature 15 to 25 degrees Celsius. Above 30 degrees Celsius the alcohol evaporates. Chemical reaction. The glycolysis pathway is also known as the EMP, Embden-Meyerhof-Parnas pathway. It degrades into a 3-carbon compound through a series of steps. Anaerobic glycolysis is carried out by organisms to produce ethanol and carbon dioxide from sugar or polysaccharides. Chemical reaction. The overall chemical formula for alcohol fermentation is C6H12O6 plus zymase, enzyme, tilde greater than 2C2H5O plus 2CO2. First stage, adenosine diphosphate, ADP, is converted to adenosine triphosphate, ATP, and 2 molecules of NAD and NAD are transformed. C6H12O6 plus 2 Namibian dollars plus 2 pi tilde greater than 2 CH3 Kaku plus 2 NAD plus 2 H plus 2 ATP plus 2 H2O. Second stage, cracking of sugar. Pyruvate is broken down into acetaldehyde and carbon dioxide. CH3 Kaku CH3CHO plus CO2. Third stage, it uses the reducing agent NAD. It is irreversibly inhibited by idoacetic acid, IC2Cu, through thiolase catalysis. NAD plus is reduced to NAD. CH3CHO plus NAD plus HC2H5O plus NAD. Let's dig into more knowledge about alcoholic fermentation. Throughout the entire fermentation process, and depending on the conditions, amount of sugar available, temperature, oxygen, etc., the type of yeast that predominates can be changed, distinguishing several phases in the fermentation. First phase, first 24 hours, non-sporogenous yeasts predominate, which resist a 4 to 5 alcoholic degree. They are sensitive to sulfur dioxide. Second phase, second fourth day, Saccharomyces cerevisiae predominates, which resists up to an alcohol grade between 8 and 16. This phase is when the maximum fermentation capacity is given. Third phase, Saccharomyces cerevisiae continues to work together with Saccharomyces oviformis. There may also be other microorganisms coming mainly from the cellars and utensils. They are usually fungi, among which Penicillium, Aspergillus, Oidium. I guess most of you want to know about the uses of alcoholic fermentation. Uses of fermentation. Alcohol fermentation has a large number of applications in the alcohol industry, wine industry, and food industry. Wine, fruit wine, beer, etc. are all products made by alcohol fermentation. The uses of ethanol in the industry are wide and range from the manufacture of cosmetic products, cleaning products, etc. There is also alcohol fermentation in the process of vegetable pickling, but the alcohol output is low, 0.5% to 0.7%, which has little effect on the main fermentation process in the vegetable pickling process, lactic acid fermentation. but it also plays a role in flavoring function.